just the GFP expression in the mesenchymal stem cells without gene jammer and with gene jammer. And we did do flow on this as well to show this. When we did use the gene jammer, we were concerned again about the possibility of detrimental effects and toxic effects on the cells. We did not see any obvious toxicity problems when we looked at PI positive cells. So these were not dead cells that we were seeing the GFP in. We did a number of experiments over time looking at the effect of the adenoviral dose on the transduction efficiency. So increasing adenoviral concentrations, obviously without the gene jammer, you saw a steady increase in GFP positive cells, but again we plateaued fairly early with the gene jammer. So this is obviously important that you can use lower doses of the adenoviral vector and get still very high transduction efficiencies. So again, this was just relative fluorescence units. Again, some indication of the level of or number of viral particles entering the cells, and remain fairly constant without the gene jammer, but at the very high end of gene jammer, you can really load up the cells with the vector. And again, we did not see any really statistically significant detrimental effects when using the gene jammer. When we're very interested in because different culture conditions can affect the differentiation of these mesenchymal stem cells, we're really interested in looking at the effect of fetal bovine serum concentration on the transduction of the mesenchymal stem cells. And even at 0% fetal bovine serum, we still saw a relatively high level of GFP expression, positive cells, and we could also go up all the way to 20% fetal bovine serum and still get good transduction with these vectors. So from 0 to 20%, we're still relatively uniform in the transduction efficiency. So we're quite happy with those results as well, because again, if you're going to use this in a cell that you're going to look at differentiating them later, you know, if you want to use with different concentrations of fetal bovine serum, or hopefully without fetal bovine serum in a more defined system. The next experiment, we looked at transduction of the mesenchymal stem cells, and now this just with with just the 85 alone fiber. So again, we moved back to just the more primitive type of adenoviral vector, and again, it did not seem to matter in the gene jammer transduction that whether it was the 85 or the 85F35 vector, we still saw very high levels of GFP positive cells with that, and again, no detrimental effects on viability of the cells. And again, you can see the different amounts of GFP expression in those cells. Okay, we did look at lipofectamine as a transduction efficiency as well, and yes, we saw some beneficial effects of the lipofectamine very similar to the gene jammer in many ways. However, we saw with the lipofectamine there was some detrimental effects on cell viability with using some of these concentrations. So higher ends of lipofectamine, we did see some problems with the viability of the cells. When we looked at differentiation of these cells transfected with the GFP, we did see that the cells, again, could differentiate even though they had the adenoviral vector transduced into them, which isn't surprising. And so, again, with the oil red, we saw positive cells as well. So these cells could differentiate again in the chondrogenic and osteogenic pathways even after transduction with these viral vectors. And at relatively high rates, again, which was very encouraging as we move forward with this work. So with the first study of conclusions, the porcine mesenchymal stem cells are poorly transduced with just the adenoviral vectors, but the gene jammer transfection had a marked increase in transduction without any toxic effects. And it's not restricted to one type of vector. It had similar results with the 85 and the 85F35 vectors. Even with the addition of approximately 10% serum during transduction, there was some improved efficiency, but again, the main thing was that you could vary the levels of serum and still get good rates. And they do differentiate into the multiple pathways. So the second study, we were looking at the effect of gene jammer, not on pig mesenchymal stem cells, but this time with human mesenchymal stem cells in rodent cell lines. And objective of this, and this was done in collaboration with a group at Baylor Medical School, to investigate the effects of gene jammer on the efficiency of human mesenchymal stem cells with adenovirus containing GFP. Again, as if the gene jammer's positive effect on transduction was dependent on the presence of the receptors. And we believe that it was an independent method, but we needed to check that out for sure. And then to investigate the effects of gene jammer on the transduction efficiency of human and murine cell lines with BMP2. So similar in many ways, we looked at different viral particle loading of the cells, and again we saw very similar effects with the human mesenchymal stem cells that without the gene jammer is fairly low at the low concentrations. If you obviously, as you increase viral particle concentration, you get a higher transduction efficiency, but again, you were able to get, actually this is reversed, sorry. The gene jammer is in the white bars, and the control is in the dark. So we did see very similar results as we had seen with the pig cells as well. And you obviously, if you look at flow cytometry for quantification, this backed up the other results we see. So the next experiment in this group of studies, we're asking, does gene jammer enhance uptake through adenoviral receptors? And so looking at various cell lines with different receptor populations, the A549, the human mesenchymal stem cells, CHO cells. So, and we looked at different viral vectors and with and without gene jammer. And the endpoint was, again, flow flow to look at the percentage of cells. So if you look at the 549 cells, high levels of the CAR receptor, the integrin, as well as the CD46, whereas you see with human mesenchymal stem cells or bone marrow-derived mesenchymal stem cells, very low levels of the CAR receptor, higher levels of the integrin and CD46, and it shows cells as low all the way across the board. So we had a varied number of cells with different characteristics as far as their cell surface receptors. With the 549 cells, there was no difference between the two vectors, or there was no difference between the control or gene jammer and the number of GFP positive cells. Again, this is expected as the 8049 cells have high levels of receptors, so the control would be very high. The human mesenchymal stem cells, again, we saw differences, again, with the gene jammer addition, and probably because of it being a receptor-independent pathway. And then as well with the CHO cells, we saw differences between the gene jammer and control on both the viral vectors. So again, this suggests in our mind that this is a receptor-independent means of introducing the adenoviral vectors into the cells. So getting on to what might be some effective gene therapy, we were looking at using the 85 expressing BMP2 and using the control, again without gene jammer and with gene jammer in the human mesenchymal stem cells or the osteoblastic cell lines. That was over a 42-hour period where they were exposed to supernate and then was quantified for BMP2. There was a cellular AP activity assay as well that was performed, and then the cells were also injected into non-skid mice for the bone assay over a 2-week period. So again, you can see BMP2 production with low viral particles per cell. Again, the gene jammer had a significant effect on the expression of BMP2 because of the high transduction efficiency with delivering it. Looking at BMP2 activity, we saw similar results again as the viral particles were increased with the 85 vectors. And so again, suggesting that this is an efficient way of introducing a vector into the animal for gene therapy. And in the bone formation over 2 weeks, you can see in the controls where the cells were injected without the gene jammer, that you saw no bone formation. But as you can see, when you have the gene jammer added, you can see the bone formation in the muscle of the limbs of those mice. So with the mirroring cell lines, again, we saw a similar effect with the BMP2 activity. And I don't know, it's a bit washed out again, but again, we saw bone formation in the gene jammer animals as well. So with the mirroring cell lines, we are also effective. So the conclusions that we have from this study is that gene jammer can significantly enhance adenal entry into the cells, and this can be obviously for human therapeutic, human mesenchymal stem cells. It's It's possible with those cells. The positive effect of gene jammer on adenoviral transduction is independent of the fiber type 5. Again, this is a receptor-independent method of introducing the adenoviral vectors. And the results suggest that gene jammer stimulates viral entry with a mechanism independent of the receptors. The presence of gene jammer during infection, the cells with the adenoviral vectors increase the expression of a functional protein, that being BMP2. So the work was done again by Pablo Bosch, as well as our collaborators Alan and Betsy Davis at Baylor Medical School. Scott Pratt was involved in this work with a company that funded this work as Biogen, and that's a company out of Austin, Texas. And with that, I'd be glad to answer any questions.
Thanks, Steve. Open the questions. Thank you. Do we have someone with microphones? Here it comes.
That's a very nice series of studies. I have two questions, please. One is, why did you choose adenovirus, and you wanted only expression for a period of time rather than constant with AAV? And my next question, if I put it together, is that's an interesting way of doing it, because what do you think? You mentioned earlier that you can influence the cell division, and a very attractive agent, for example, is HTERT, which could enhance the cellular renewal in a terrific fashion. But we were very interested in that, and then we had about one paper showing that it actually produced malignant transformation, and probably after many times or many divisions, and your strategy might get over that.
Yeah.
Your thoughts, please. Why did you use with EMP2, why do you want it for only a period of time?
Well, this is really a proof of concept type of study, and I didn't show the data on the AAV vectors that were used with GFP, but they were positive. I mean, very similar to what we saw with the 85 and 85 F35. So we could, with the gene jammer, get very high rates of transduction as well as stable integration. So we could select with the AAV, we could do flow cytometry and FACS and sort out the cells and do clonal derivation of cell lines with that. And I just didn't have enough time to show you that, but it works just the same across the board for any of those. And obviously, if we want stable transfection, that's the way to go. There are people, and you know better than I do, that are suggesting that there may be some things that you'd want short-term expression, and that's why you would use that, obviously. But I've also had questions, well, does it also work with lentiviral vectors or other vector systems, and we just haven't tried that as yet. But for BMP2, that's the vector that the people at Baylor had available and were interested in. I don't know, it's a good question. I should ask if they had the AAV version of that, and probably be worth trying as well. But the AAV does work with gene jammer, and you can get clonal selection of those cells.
HTERT?
HTERT. Yeah, that would be of interest for expanding the cells over time. Trying to think. you know, there have been a few groups that have put in H-TERT into cells and fibroblast cells, and yes, they work to some extent. I know that a group that's, I'm trying to remember the name, in North Carolina, that they don't believe that H-TERT alone will be sufficient to get long-term expansion of cells, that you probably need some other things like a large T antigen or something like that that you'd transiently be able to express from time to time that would be useful. So, but I think, yes, H-TERT would be a really nice one to put in and expand.
May I ask another question?
Please.
Bad publicity about adenovirus.
Mm-hmm.
Is that going to last with us, or was it a dose effect, or
Yeah.
have we seen the end of it?
I'm not I'm more of the stem cell person than the, so I'll, anybody else that would like to have their point of view put across, I don't know. I mean, you know, there are companies still out there, Genbeck, that is still very interested in some uses with VEGF and some other things for adenoviral vectors. They haven't gone out of business yet.
Bob Goldberg over there. And then Robbie.
Thanks, Steve. That was great. My question is, does it need to be mesenchymal stem cells to do this with skin fibroblasts?
Yeah, I'm trying to think back. We did do some with the skin fibroblast cells, and it worked just as well. So again, it's not a receptor mediated, so it's a pretty homogeneous across all the cells that we've tried. But we have tried with the skin fibroblast cells, with the pig skin fibroblast cells, and then we used them in the nuclear transfer procedure and produced some transgenic embryos and that was with the AAV system.
Yeah, I had a similar question. I was wondering what your thoughts were on using muscle or this muscle and
I think it should. Right. Yeah, I think it should be tried. We didn't try muscle cells, but I think it's a robust system to get the adenoviral vectors in.
Are there other questions for Steve Stice? If not, thanks, Steve. We'll move on to our next speaker, Pat Staten, who, since he's spoken before, doesn't need an introduction. And this time, you've done everything possible to not have a 30-minute computer malfunction delay.
It's not working. Yes. So I want to thank Steve for, I didn't realize how great that would be as a talk right before mine. I'm going to take a step back and talk a little bit more generally about how nucleic acids are currently delivered. But I'm going to focus on viral delivery methods. Yesterday in my talk, I tried to put protein delivery in the context of tissue regeneration strategies. And again, then just to quickly summarize, a lot of times what we're after is controlling events within the scaffold. But we always have to remember that when these go back in vivo, that there are also interfacing events that we have to control outside the scaffold. And so you really need to think about being able to control cell behavior within the scaffold as well as outside the scaffold. And so today I'd like to just add into the repertoire of methods that we can use to control behavior both within and without the scaffold, nucleic acid drugs. And again, these have been used and investigated to control cells as they come into the scaffold. They've been used to control cells, as Steve was just going over, even prior to the point in which they might be added to a delivery vehicle, a cell delivery vehicle. But you can also think about these, and I'll give you an example of how nucleic acids can be used to control events around the scaffold. And these, again, probably most prominently would be targeted toward the control of angiogenesis in the foreign body reaction. There are, for the most part, 3 general categories of nucleic acid drugs that are under development. The first is, in fact, genes, so that would fall under the category of gene therapy. The other 2 categories that are being investigated are antisense drugs, which are short pieces of synthetic DNA, modified DNA usually. And antisense therapy has kind of been a roller coaster, up and down, and people have very strong opinions about the likelihood of whether these are going to be a real therapeutic, but still under active investigation clinically and through several companies. The sort of new kid on the block that is taking all the headlines is siRNA, which arises from a recently discovered biological phenomenon. I'm always amazed still in biology that we can, still in this day and age, discover incredibly new biology. And RNA interference has just been recently, relatively recently, discovered, but there's already a huge investment from the private side of things into this technology. So people are trying to think about how we can use RNA interference through the molecules being siRNA. But front and center is how do we deliver double-stranded RNA in this case as an actual drug. There are 2 broad strategies by which you can deliver any of these 3 nucleic acid drugs. Steve went over viral delivery, an example of viral delivery, and then the other broad category is non-viral delivery. And there are a lot of advantages and disadvantages to both approaches. Both are potentially viable at this point in our evolution of tissue regeneration and tissue engineering. And so within the context of a within the context of tissue engineering, what we're talking about are coming up with strategies to incorporate these nucleic acid delivery agents into scaffolds. And again, that has been done with both viral delivery and non-viral delivery. There are so many options available here. But again, the broad categories fall into viral delivery versus non-viral delivery. The simplest non-viral delivery technique is to use bare DNA. And this, in fact, has been done, and it's been the basis for startup companies. I would say most of the effort and current investment is in synthetic vehicles, and this is where I'm going to focus today. There's likewise an entire field of different types of viruses that can be used to deliver, and I'm just not going to focus on that. From the standpoint of delivery, this is where the big challenges are, is to see if we can raise up our level of efficiency with non-viral delivery systems to anything approaching the efficiency of biology. And So let me start with gene delivery, although this also is a strategy for delivering antisense and siRNA, and that is with the most basic form of these drugs, which is a plasmid. And I'm not going to go into the biology of this, that's sort of for other people, I guess, to cover, but there really is a lot of design and biology that goes into the design of the plasmid. And while the gene is probably the part that you would think about as the drug, the details of the plasmid construct are very important. And so they usually contain a promoter, the actual gene of interest, and an antibiotic resistance gene. But there's been really huge advances in that come from the molecular biology side of things in preparing enhancer-specific promoters for longer-term expression and for tissue-specific expression. And I don't want to gloss over that. It's just sort of outside the scope of this talk. What I'm going to be talking about is once you have in hand the right construct, how can we more efficiently deliver that construct? The reason why this is such a challenge from a drug delivery perspective is that there are barriers that go across the whole organism down to the details of intracellular trafficking. And at all levels, there are significant barriers to getting a functional gene to the right place where it can do its action inside the cell. Outside the cell, at the level of the whole organism, there are huge challenges and huge unknowns, really, when we take, for example, a DNA plasmid and put it into either a systemic delivery or a local delivery environment. There are serum components that can bind to DNA, sequester DNA, and this can be either a positive or a negative depending on the details. Sometimes it's a positive to sequester the DNA, it can be protective to the DNA, but it alters then the bioavailability of that plasmid. It affects which cells can pick it up, it affects where it goes even in the body. Likewise, there are nucleases around. This is an obvious problem that can degrade the DNA. When we get down to the level then of getting within target cells, the DNA obviously is going to have to get to the nucleus eventually. But there are many different pathways by which the DNA can be picked up by the cell. Once inside the cell, it's not truly inside the cell, as I'll go into in a little more detail. It's picked up in a vesicular trafficking pathway, and how do you get that out of the vesicular compartments to the cytosol and then to the across the nuclear membrane barrier. These are, from a drug delivery standpoint, daunting challenges, especially when you begin to add them up. There have been two major approaches then to dealing with these barriers. The first is to use cationic polymers to condense the negatively charged DNA. And because these are polymers, the resulting condensed particle is called a polyplex. The other major approach is to use a cationic lipid, and sometimes this term liposome is used. I have it on my slide because it's used so much, but it's not actually a very accurate description of the structures of the resulting complex. It's not very well described exactly what the structure is, but probably not liposome. Nevertheless, the cationic lipids have been the other major category. They share in common the positive charge that interacts electrostatically with the negative charge on the backbone of DNA, and the condensed particle in this case is called a lipoplex. And so you'll see lots of different companies out there that are trying to develop polymers and polyplex technology, and another category of companies that are working on cationic lipids and lipoplex technology. The key point is that both of these agents, when they condense the DNA, they sequester the DNA and protect the DNA against nucleases. To some extent, if they're designed appropriately, they can also protect against serum inactivation by the binding of different serum components, although that remains a serious problem and detail that you have to worry about in the design of either your cationic polymer or the cationic lipid. And then, in addition, they condense the plasmid DNA into very small particles, typically under 200 nanometers in size. Some people say that you need to be under 100 nanometers in size, but the general point is that the condensation allows the DNA to be taken up with much higher efficiency. Looking at the cationic lipids first, there is a whole literature on different cationic lipids, and there's a whole variety of biotech companies that have their favorite and protected cationic lipid compositions. The main game here is to get efficient condensation without toxicity. And this has been a major issue with the cationic lipids, is toxicity. In vitro, this is maybe a little less of an issue because you can take some hit in toxicity and still get your targeted cell population. But in vivo, this has been a major problem and a barrier to the clinical development of cationic lipids. One interesting point in regard to this that is, again, part of the challenge of doing this with high efficiency is that on the one hand, the first thing that you want to do is condense your DNA very tightly so that it's sequestered from nucleases and from serum components. But you have to realize that once it gets inside the cell, it has to decondense so that the DNA and plasmid can get to its target and be in a form that's accessible for transcription. And so these two design criteria go exactly opposite to each other. On the one hand, you're wanting to condense very tightly, but in the other case, you're wanting to decondense. And there's major effort to design into these systems degradability so that it condenses outside the cell, but through some mechanism, reduction or cleavage, once inside the cell, you can decondense the DNA from the degraded carrier. This is a cartoon of in this case, sort of a liposomal cartoon of how these one of these cationic lipids can be used to condense DNA. And so you're seeing particles again on the size scale of about 100 nanometers. This should be viewed purely as a cartoon. The actual structure of these complexes it has not been so well described, and it's very dependent on the type of lipid and the ratios of the lipid to DNA. And in my opinion, this is a detail that needs to be considered because it plays a big role in the decondensation process that ultimately has to occur inside the cell. This is a cartoon for the polymer, cationic polymer approach. And I guess Ringsdorf really was the first to propose this kind of picture, this advantage of polymers as carriers. The nice theoretical advantage of polymers over cationic lipids as a carrier is that you can build in additional functionality. And so you can have the cationic charges along the backbone that condense the DNA, but by relatively simple grafting approaches or incorporation of specially designed monomers, you can add additional functionality. And some of the most obvious things that people have been trying are to add in peptides that represent nuclear localization sequences that can address that barrier of entry from the cytosol to the nucleus. There can be additional agents that act to get the carrier, once it's taken up into the vesicular trafficking pathway, get it out of that pathway, usually from the endosome. and thus get over that barrier of entry to the cytosol. You can also add in targeting agents that stimulate uptake more efficiently than pinocytosis so that you can get receptor-mediated endocytosis. And you can also think about adding in PEG groups, are the most common groups, that sterically stabilize the complexes against serum inactivation. And this has been a huge problem, this serum inactivation for the cationic lipids, but it's been shown now, I think, and there is a lot of literature and clinical experience now with pegylated polymers that show that this can be a very important component in aiding the escape through the reticuloendothelial system for systemic delivery, or locally to control nonspecific uptake of the particles if you have a targeting agent that can target the appropriate cell that you're after. A couple of examples, the sort of first generation polyolysine and polyethyleneamine are examples of cationic polymers. And in fact, most of the work has been done with simple cationic polymers that don't have the more sophisticated elements. Polyolysine is not really a viable candidate anymore, I think. For in vivo work, there's a lot of toxicity associated with that composition. PEI is still used a lot, and there are a lot of names for these different companies, like Gene Jammer that Steve was talking about, the different companies that produce silencing RNA have their own little names, but a lot of these polyamine polymers are in fact modified versions of PEI. And PEI, the details are very important. It gives you one idea of how you can get these structure-function relationships going on the delivery side. There are highly branched versions of PEI versus linear versions. That branching versus linear has been shown to be an important detail in their efficiency, but also their toxicity, and it's usually a trade-off between those two. And also molecular weight has been shown to be very important, both from the standpoint of toxicity and efficiency. But this is really certainly a very active area, both in academia and in industry, is trying to come up with better polymer compositions. With antisense, moving to the other two types of nucleic acid drugs, basically antisense is a method for inactivating a specific mRNA species at the mRNA level. And so it basically just interrupts the central dogma at the mRNA stage. And these are short pieces of DNA that hybridize through complementary base pairing to target mRNA sequences. They're usually chemically modified, so they're not as susceptible to nucleases. This is one of the nice things about antisense drugs as compared to plasmid-based drugs, is that you can conveniently chemically modify them. That chemistry has been pretty worked out at this point in development. There are a few competing modification chemistries, but they're out there. And by this hybridization, then they mark the mRNA for degradation by an RNase. And there is one approved antisense therapy drug. It's an antiviral drug. It's not a big seller, but there has been one that has been FDA approved, and there are many more under development at several different companies. The really exciting new drug, for which there's been just incredible capital investment in the States, takes advantage of this process of RNA interference. And I'm just going to sort of point out here, I guess, that this is an RNA-based drug that has in common the ability to target and degrade a specific mRNA sequence. And so it has that in common with antisense therapy approaches. these companies, such as Alnylam, which is one of the biggest ones that was formed initially around this biological phenomenon, it widely recognized from the beginning that delivery is going to be a big issue with RNA. And again, there are methods for chemically modifying RNA, but in this case, it's still a little uncertain what types of modification and how much modification you can get away with, because there is a very specific biological system that these drugs have to utilize to affect mRNA degradation. And it's still a little unclear how modifications of the siRNA are going to affect that process. Now, it has been shown, though, that stabilizing nucleic acid modifications can still allow function, but it's still relatively early. The approaches are basically the same for shorter DNA segments, oligonucleotides, for antisense, and for silencing RNA. There's the same cationic polymer approach to condensing the nucleic acid, smaller nucleic acid drugs, so you get polyplexes, and also cationic lipids for lipoplexes. And so the delivery technology looks very similar at this point from this general standpoint. In summary, then, I would say that early clinical experience with these non-viral delivery methods has been quite disappointing. Several of the companies that were formed around the new cationic lipid technology, as well as polyplex or polymer-based technologies, have gone by the wayside and had disappointing clinical results. The basic reason is that biology is really hard to beat. Conservative estimates of the difference between analogous viral delivery methods, so say for the same plasmid or gene, versus non-viral, are better by 10 to the 6th. I've even seen 10 to the 13th demonstrated in the literature. I mean, that's a huge number that you're up against. And so at this point, a lot of people are sort of wondering, are we ever really going to have non-viral delivery methods that find their way to real-life products? The reasons, again, are because there's all these barriers, especially from a systemic delivery standpoint. When you're talking about these biomolecular drugs, there are huge issues of stability, transport, how they get picked up in various places. They have very poor pharmacokinetic properties. Targeting, how will they ever find their target cell and get inside? These have to get inside the cell, that's a key point. And so intracellular delivery has really been shown to be a huge barrier in vivo. Even if they get to the right target cell, getting a macromolecule or a nanoparticle inside the cell is far from trivial. Getting it to the right place inside the cell is what's really not trivial. So I'd like to just close with, to give you an example from our own group of, I think despite having said all those negative things about what we find interesting, number one, it's a challenge that if you're sort of on the delivery side, you just can't get away from it. It's just such a big challenge. There's such a need for it. But where we got interested was actually from thinking about the good news. And the good news is you always have to remember that biology has figured out a way to do this. And so there is a design example out there that we can look at. And if you begin to look at it in more detail, you don't really think anymore that these viruses are simple organisms. They're amazingly complex and they're amazingly efficient. And what they really are built to do is to deliver macromolecules to the right place inside the cell. That's really what they do in an unparalleled way. And what probably is well appreciated, and Steve was talking about this, are some of the details by which they very efficiently enter the cell through receptor-mediated endocytosis. But what I think is way less appreciated is that many viruses and many pathogens, so protein-based pathogens such as diphtheria, cholera, share this common challenge of the fact that when they get taken up, they're still not inside the cell. They're in a membrane-bound compartment called the endosome. And the problem with this, if you're a macromolecule, biological macromolecule, is that this gets evolved into the lysosomal compartment where they are degraded, or they get recycled back outside the cell. And so what pathogens have evolved is a delivery system that's activated inside the endosome, and it's activated by the fact that these endosomes develop a low pH gradient. They go all the way down to pH 5 to 5.5. And they have a protein that converts at low pH to a membrane destabilizing form and activity. And so only once they're inside the cell, inside the endosome, is this protein activated to destabilize the membrane to aid the transport of their drug, be it DNA or protein, to the cytosol. This has been studied in some detail, for example, in the flu virus that is schematically shown here. There's a protein called hemagglutinin that is activated. At pH 7.4, it's in a stealth-like conformation. It has no activity, membrane activity. But as the pH drops inside the endosome, so down here would be inside the endosome, as the pH drops, there is a triggering of a large conformational change in this protein, and it exposes side chains that partition into the endosomal membrane and destabilize it. And in this form, then this membrane destabilization allows the effective transport of these relatively large particles across that membrane into the cytosol. And it's been studied in some detail, but this is the question that we got interested in. Could we mimic these biological machines with safer synthetic systems? And so what we would be after is the synthetic analog to these viruses that can enter into cells efficiently. That part is fairly easy. But have this additional activity that synthetic systems really haven't had in, as the pH drops in a reversible manner, the delivery system would destabilize the endosomal membrane and get the nucleic acid drug to the cytosol. In a little more detail, the mechanism of hemagglutinin is described even better. There is a very specific glutamic acid side chain with a carboxylate residue, and it's the simple protonation of that side chain that triggers this large conformational change in the protein. And it's because the pKa of the carboxylate is tuned to this pH gradient that occurs in the lysosome. The pKa can be tuned by the residues around it to that region around pH 5.5 that these pathogens find in the endosome. And so they go between a hydrophilic, stealth-like conformation to a hydrophobic alpha helix that is membrane destabilizing. And it's very important to recognize, because this is a protonation-deprotonation event, that this is reversible. And this is how the pathogens accomplish this without being particularly toxic, in many cases, to the cell. Once it goes back to the cytosolic pH, then it becomes this hydrophilic polymer again. So we were after a synthetic equivalent. I'm not going to go into a lot of the chemistry, but you can see that there's two key pieces to our design. There's a carboxylate residue that is the same pH sensing unit that's found in these pathogenic systems. But then we have alkyl segments, both within the same monomer, but also can be provided in different monomers. And it's that alkyl segment and the details of that are related to the membrane destabilizing activity. It's these groups that, when this carboxylate becomes protonated, partition into the membrane to destabilize the lipid bilayer packing, and then that creates pores, temporary transient pore formation that larger molecules can exploit then and transport. And then there's a carrier monomer here by which you can add in proteins, oligonucleotides, siRNA, and I'll show you an example of that. We've shown in a lot of studies, this is a recent illustration, that these polymers do have this property of a pH-dependent membrane destabilizing activity that is reversible. And so in this assay, this is an assay that's been used for many years in the field, a standard assay, standard conditions that allows you to compare different compositions across the literature. And it's an outside-in membrane destabilizing activity. You add polymers to a suspension of red blood cells, and then as a function of pH, ask the question, how effective are they at destabilizing that membrane sufficiently for hemoglobin to come out of the cell, which is what you actually measure here on the y-axis. It's called hemolysis. And so I'll just show you that at these compositions, at pH 7.4 physiological pH, you have very low membrane destabilizing activity. But by the time with this composition you're up or down to pH 5.8, this polymer is converted into a membrane destabilizing form. And we've done a lot of work. You can very nicely tune exactly what pH this occurs. We have polymer compositions that will destabilize at pH 6.6, for example, and not at 7.4. So these are activated extremely sharply, very similar to the biological proteins across a pH range of less than a half a pH unit, and it's fully reversible. So the idea then is that we might, for example, through this monomer add a cationic graph that condenses either negatively charged plasmid, negatively charged oligonucleotide, or negatively charged siRNA. And the active form then is a polyplex, or a condensed particle. And the details of size and charge are very important, and they can be controlled very nicely in a rational way by the molecular weight and the composition here of the polymer. But just to emphasize, these are particles. This is one composition, dynamic light scattering studies, to show you that the size range is around 200 nanometers for this particular condensed form. But just to emphasize that these do form particles. I'd like to just give you one application, and that is in the tissue regeneration area, is control of the foreign body reaction. Shown here, the response to a scaffold. There's been a lot of discussion of this. And we've been very interested through UWeb with Buddy in trying to control the foreign body reaction by a nucleic acid approach. And our approach targets the macrophage, which has been an incredibly difficult cell to get nucleic acids to, because it has a form of the lysosome that's generally called the phagosome that is particularly rich in these nucleic acid degrading enzymes. What we're after is signaling pathway modulation, so not to simply knock out the macrophage, but to very specifically control the activation state of the macrophage. You don't want to knock it out. There has to be some inflammation so that you can enter into wound healing. And so we've been conceptually interested in the idea that you can, in a time-dependent way, knock out very specific signaling pathways that are associated with the macrophage activation. And let me show you the exact composition here. Similar composition, there's a membrane-disruptive polymer backbone that is pH responsive. In this case, we've just grafted on through a disulfide linkage a polylysine segment that electrostatically condenses antisense oligonucleotides, or siRNA drugs. And this disulfide allows the carrier to separate from the drug in the cytosol. The key new thing, really, again, just to emphasize, is that the polymer has an activity. It's not a simple carrier. The target that I'll show you some data for, the mRNA target, is interleukin-1 receptor-associated kinase. This is a key kinase that is constitutively expressed and is up very near the toll receptor pathways, but also the IL-1 beta pathway, which is very important for the foreign body response to biomaterials. And And so the idea is to knock this out, at least transiently, by delivery of an antisense or siRNA against that IRAC mRNA. This is IRAC1 for the detail. But essentially what you do by knocking out IRAC is short-circuit the macrophage's ability to receive pro-inflammatory signals. And you can measure that if you, for example, challenge macrophages that have been treated with your drug with LPS. Is it true that, for example, you lose the connection to the upregulation of TNF-alpha? We've shown that. I'm just going to show an siRNA result. We can do this in vitro, sort of, with any number of different antisense and siRNA drugs. This is kind of a typical result. Here what I'm actually looking at, rather than TNF expression, is by RT-PCR actual IRAC message levels. So if you have no carrier, this particular siRNA has no activity in the macrophage. And we've just found that to be generally true. It's hard to get nucleic acids into that target cell. This is a dose response then as we're delivering with this pH-responsive carrier, and you can see a very nice knockdown in a dose-dependent way. And you can do a similar study, maybe not as effective. It is, in our hands, we've seen better siRNA results than with the corresponding antisense. And I'm not going to go into details, but we actually have had these in a number of animal models now and shown, for example, in a wound healing model at UWeb, that these types of pH-responsive carriers are really, it's kind of an all-or-nothing approach in this local delivery system with a plasmid. And they've also been in a lot of systemic delivery models to look at toxicity and biodistributions. And they pass sort of the first test, but there's really a lot more preclinical validation that needs to occur in specific applications. But I give this sort of as a specific example. I could show you several others from different groups. This is an area that's very active to try to build more active polymeric delivery systems. And I use that to illustrate the point that what people are after at this point is something that goes beyond a simple carrier, because those just have been shown to be insufficient in the clinic. And so these new delivery systems have some sort of activity to get around these barriers. And in our case, the primary technology is endosomal escape and this property of the polymer itself in a pH-dependent way being membrane destabilizing. But of course, in addition, they have the usual advantages to condensing, to providing serum stability. I didn't really point that out so much, but these polymers also provide a lot of serum stability, so that compared to most cationic lipids, they show much higher activity in the presence of serum. And so for in vivo applications, this is a key point, obviously. So with that, I'll just close. I guess maybe you can see, I was thinking this morning when I was looking at this, this is our pyramid, I guess, in Seattle. It's called Mount Rainier. The disadvantage of our pyramid is that it can blow up at any second. So I don't think it's quite as stable as your pyramids seem to be. But thanks for your attention, and thank these people over here for funding.
Thank you, Pat. This presentation is open to questions.
May I ask a basic, probably naive question? There has been general neuroelegant lecture about the modeling of the nucleic acids. I mean, it's a crucial point in non-viral delivery systems. On the other hand, protein modeling has been the floor of considerable progress regarding the software. How far we are in the area of modeling of nucleic acids? Can you elaborate on this, the software, how the three-dimensional configuration of the DNA or the RNA?
Well, I can't answer it with any detail. I mean, to my level of knowledge, structure of DNA is so well defined conformationally that it is, in my mind, it's pretty well described. With siRNA as well, there's some initial high-resolution structure analysis that have been done on some of those forms, and they adopt typical nucleic acid, helical conformations. But when maybe what you're referring to is some of the details of like single-stranded DNA conformation, or also within the complexes in the condensed state, what would be the conformation? And there, I don't know about modeling. I know more about a few attempts to experimentally look at the structure of those complexes. And there, the answer has been very clear that it's not a liposome-like structure, that you find many different lamellar-like structures and phases that can be found in any sort of condensed lipid. Phase is probably the right word.
Go ahead. Yeah.
In SSCP, for example, or TGE, or whatever, we capitalize on this. Specifically, may I say, if the liposome, if you pack inside the liposome 20 base pair antisense, or is 23, and 23 with different one base pair.
There's a size range, yeah.
It doesn't depend on the three-dimensional configuration of the nucleic acid molecule itself, or is it?
Well, see, usually it's because there's an electrostatic interaction and a condensation of the DNA. So it's not well viewed as an independent DNA molecule that's sort of encapsulated by the carrier. Now, there are examples of that. But for the most part, when you're talking about a cation and a carrier that electrostatically condenses the DNA adopts a very different conformation, a condensed conformation, compared to, the double helix, extended double helix that you're used to looking at. And so, certainly with these types of carriers that I've been talking about today, you're in a condensed form with the carrier. No longer does that simple picture apply that you have an independent nucleic acid molecule with a surrounding vehicle. The two interact into a new condensed phase.
But regarding the naked DNA, what about the naked DNA? I mean, the way that you inject naked DNA.
Well, that again, so yeah, if it's like a plasmid, it can be, you can talk about supercoiling and of course structural properties like that, but single-stranded DNA, because it's like a negatively charged polymer, it's an expanded, almost random coil type of conformation that doesn't have a really well-defined atomic level structure.
Maggie, did you want to make a comment?
Yeah, just a quick comment. Beautiful talk. What's the place of combining several strategies, viral and non-viral, like something like hemagglutinating virus of Japan, like also?
Yeah, those have been kind of fun. I've found those fun, and actually Steve's talk was interesting. There's something there as well when we're trying to add together. I think what those polyamines are doing is a fairly well-described mechanism that Jean-Paul Baird has described in detail, the proton sponge effect, which is probably the mechanism by which those polymers provide some destabilization of the endosomal membrane, and that's presumably how they enhance transfection. But people have gone way beyond that, and it's very interesting. They've been synthetically modifying viruses. And you can definitely think about this. We've had several people approach us with the idea of trying to somehow chemically combine our synthetic polymers with the viral particles themselves. And I think that's kind of a fun area to think about, is trying to combine the two. I guess I hadn't really appreciated so much this problem of the right receptors not being around with the viruses.
Well, I think we need to move on. Thanks very much, Pat.
Good morning, everybody. Before we get into the next speaker, I would like to acknowledge the effort, the endless effort done by the National Research Center to keep us all here comfortable and happy and to get everything on schedule. And I would like to acknowledge the effort done by Dr. Osama Shabrawi, Dr. Mona Toki, Dr. Magda Ramzi, and Mr. Ashraf, Mr. Ahmed, Mr. Hatem, and all the people that they have. Did I forget? Dr. Yahya, that's for sure. Dr. Yahya Zakaria, that he has been working for a whole year to get the schedule going, which I see the effort done here. Thank you very much all for this. Thank you. Our next speaker is Professor Mustafa Al-Awadi. Dr. Mustafa Al-Awadi, I know him for 10 years now. The first time we've seen each other, I think it was in Genetic Engineering Institute at Mubarak City. And I was lucky enough to supervise master's thesis in tissue engineering along with him and other eminent professors. And after that, we came close to know how far Dr. Mustafa Al-Awadi is doing in virus hepatitis. He's very well known with his activity in it. And I know he has several patents, I know that for sure. And he accomplished a lot to the field of virus hepatitis, especially in Egypt. Dr. Mustafa Al-Awadi will speak to us about, he's the head of the Division of Human Genetic Engineering at the National Research Center here. Has a lot of departments under his supervision and a lot of numerous studies and supervised many researchers under his direction. Dr. Mustafa Al-Awadi will speak about a very hard topic here for me. Would you be kind enough to pronounce it, please, sir?
Good morning, everybody. Thank you, Mona, for this nice introduction. My talk is going to be not very sophisticated in terms of the delivery systems, but probably it may cover the part that was preliminary to what Pat Steichen talked about. Pat Steichen talked about all the ways to deliver DNA. But I'm going to talk about how to design an antisense molecule to stop the replication of hepatitis C virus and what are the data that we use to depend on to develop these antisense molecules. And the rest of the story should be completed by Pat Steichen's work. And I thank him very much for all the elaborations he gave about the different delivery systems for the antisense and the small interfering RNA molecules. The reason why I used the HCV for my talk is that HCV is a very big health problem in Egypt and all around the world. It almost affects more than 15% of the Egyptian population, and the current standard therapy for HCV, which is the interferon, is unfortunately disappointing. However, after the pegylation of the interferon and the addition of another drug, which is the ribavirin, so the pegylated interferon plus ribavirin has also been disappointing. The rate of recovery did not exceed 50%. So this has become an enough motivation for many investigators in this country and in other countries to try to develop alternative therapies for HCV. So the very complicated title is just the phosphorothioate oligodeoxynucleotide. We can tentatively call it as antisense molecules or antisense drugs. I'm going to give you an introductory slide about the natural history of the HCV infection. This infection is a very long life infection. It may cover almost from 20 to 30 years from the patient's life. It starts with the acute infection that may last from 4 to 6 months, and then 80% of those infected with hepatitis C will develop chronic infection. And this chronic infection stage will last almost for 20 years in males, and it may last for longer period in females. Part of these patients, or approximately 20%, will develop hepatic cirrhosis with a high risk of developing hepatocellular carcinoma at a rate of 1 to 4% from the patients every year. Most of the patients with HCV are residing within the chronic hepatitis state. So when you see a patient with hepatitis, chronic hepatitis infection, most probably he will be in the phase of chronic active hepatitis state. Among these groups, there was a very interesting group of patients that we call the healthy carrier, tentatively called the healthy carrier. And these patients may represent about 30% of the patients with chronic hepatitis. And they are really healthy. The only sign that they have hepatitis is that they have hepatitis virus RNA in their circulation. Otherwise, they have normal liver histology by all means of pathological examinations, ultrasound, and everything. They have normal hepatic enzymes. They have constantly low viremia, and they have no extrahepatic manifestation. The most important is that they have almost none, or at least reduced core antigen level. The core antigen of hepatitis C virus is the major pathogenic protein of the virus that has been shown to be involved in the hepatitis, steatosis, cirrhosis, and also recently the transgenic mice with core antigen could develop hepatocellular carcinoma within about 45 days of their lives. So which means that the core antigen level is a real pathogenic indicator, or the real indicator, biological indicator for the hepatitis C infection. So what are the factors that may influence the severity of the HCV-related liver disease? These factors might be viral, host, or cellular. Viral, by definition, is those mutations that happen due to the lack of fidelity of the HCV RNA-dependent RNA polymerase, and the mutations, especially in those genes that are known to be phylogenetically conserved among all the isolates of hepatitis C. Any mutation within these highly conserved regions may have influence on the life cycle of the virus. The host means this endocrine derived or driven differentiation between males and the females, the immune composition or the immune makeup of the patient. Cellular means those transactive, lots of transactive factors, nuclear transactive factors that interact with the viral genome and make up the entire phenotype of the HCV infection. So we will focus in this talk on the viral factors, and we will try to derive the important sites of, or the important sequence within the viral genome that will help us to design a suitable antisense molecule for as an alternative therapy. So this is the HCV genome. It is tentative. The HCV genome is a single strand RNA of a 9.6 base, kilo base, and the genes within the genome encode for either structural or non-structural proteins. The structural proteins include the core and the envelope glycoproteins. The non-structural, the genes that code for the non-structural proteins mainly encode for the group of enzymes related to the viral replication, serine protease, helicase, and RNA-dependent RNA polymerase. Due to the lack of fidelity in this enzyme, lots of mutations are encountered, particularly in the envelope and the core. And this is for reasons for the virus to escape from the immune stress of the host. However, there are very highly conserved regions, which are the regions coding for the viral replicating enzymes, and also both the 5' and the 3' untranslated regions. May I have another pointer, please? So these 5' and the 3' untranslated regions are characterized by that they are very highly conserved among all the reported quasi-species of hepatitis C. And the reason of the conservation may be related to that the structure is highly structured, and it contains lots of secondary structure with stem loops around here, and these stem loops triggers the potential binding with the transactive factors. Oh, so this is fine. This is fine. This is fine.
He likes Pat statements.
Yes. So this is a magnification of the 5' upstream region, which is also called the iris region, the internal ribosomal entry site, because it is the major region of the virus that is related to the initiation of viral protein translation at the host ribosomes. The most important of this secondary structure is the initiating methionine, and this is the initiation of the core gene. So the AUG start codon is here. And when we go upstream towards the 5' and the entire length of this is 340 bases. And this secondary structure includes a 3D structure, or we call it the third complex structure. And it can be termed as 3A, 3B, 3C, and 3D. And this 3D is one of the most important secondary structures of the iris region of hepatitis C genome. The S1 nuclease protection assay has revealed that this is the 3A, 3B, 3C, and 3D. So the eukaryotic initiation factor 3 may bind to these loop structures, and it regulates the initiation of translation, and also it regulates the expression of viral proteins. The 40S ribosomal subunit and the 5S ribosomal subunit will bind to the loop structure at the 3D complex. And this triplet G around here so the 40S ribosomal subunit protein and the 5S ribosomal subunit protein will bind to the loop structure around here. And it is very important to have a triplet G at the apex of the loop structure. Site-directed mutagenesis studies have revealed that elimination of any of these Gs will dramatically knock out the binding of 40S subunit protein to the loop structure at the 3D stem loop. And eventually it will affect dramatically the translation of the polyprotein precursor of the virus, starting from the initiating methionine, and the viral proteins will be dramatically reduced. So going back to the patients that I have described in the beginning, who are termed as healthy carriers. These patients, we tried to analyze why they are healthy. Probably their immune system was different from the immune system of the patients with aggressive liver disease. And probably the viral quasi-species infecting these people have dramatic mutations that affect the viral replication. So we decided to analyze the sequence at the upstream of the 5' upstream region. And particularly we concentrated on the 3D stem loop structure in our patient. So this is the triplet G that is very important for binding to the 40S subunit. And by analyzing some of these patients, we found some of these patients encountering mutations, just single or double mutations somewhere. So this is an example of these mutations. A GC has been converted into AG. And what happened is that the 6 loop, the 6 nucleotide loop has been, by the electronic flow, has been changing into an octa loop or octanucleotide loop with a partial shuffling of the triplet G to be not in the center or at the apex of the stem loop structure, which with a possibility of failure of binding of the ribosomal subunit and the consequences of reduced viral replication and reduced gene expression. So this is the type of dinucleotide mutation that we found in one of the healthy carrier patients. And we have analyzed the stem loop structure in a number of these patients, and we found that about 50% of them have encountered mutation in this highly conserved region. So, but the take-home lesson from this particular mutation is that the triplet G is very important and significant for the viral replication. So from this point, we started off trying to design an antisense drug or antisense molecules and to test whether these antisense could inhibit viral replication in vitro. So we decided to design 2 antisense molecules. One of them is our control that was designed against the initiating methionine, and the other one was against the 3D structure, including the very important phylogenetically conserved triplet G. This cartoon will show, I think Dr. Steiton showed us how these antisense molecules work. It blocks or it interferes with the translation machinery so that the RNA cannot be translated, and also it triggers the intracellular RNase H that degrades the RNA involved in the heterodimer between the short DNA molecule and the RNA of the virus. So we wanted to establish an in vitro system to propagate HCV to study the effect of these antisense molecules. So most of the people use the transfection experiments, in which they use either full-length HCV cDNA, or they in vitro transcribe this cDNA into a viral RNA, or they use subgenomic HCV fragments and they link these to a reporter gene with a luciferase activity that can monitor the infectivity of the replicon into the cells. However, we found that this strategy has some disadvantages. First, it is not permissible to the viral replication, and because it starts with a cDNA, it lacks both the 5' and the 3' non-coding regions, which is the focus of our study. And also, due to the lack of fidelity of the RNA-dependent RNA polymerase of the virus, it incorporates lots of mutations with a chance to incorporate artificial splicing sites, so the RNA is sensitive to intracellular spliceosomes and the processing, so degradation is also possible for this molecule. So we decided to go back to the natural way of infecting the monolayer cells with HCV positive sera. And we had options to use hepatoma cells or hepatoblastoma cells or fetal hepatocytes or fused primary human hepatocytes with hepatoblastoma cells. We decided to use the HG2 cells because that they have more than one advantage. They are the closest of the cell lines to the normal hepatocytes. They have the LDL and the CD81 receptors, which many investigators reported that these receptors mediate the entrance of HCV particles into the cells. Most importantly, it has been reported from several labs the presence of a 66 kilodalton membrane protein on the membrane of these cells that act as a receptor for the antisense molecules. So it is good for our particular purpose. These are the HG2 cells, and these are the antisense that we designed by the board. This is the SODN2 and this is the SODN1. This includes at the beginning of the core gene, so this is the initiating methionine, and this is the triplet G at the apical part. It is highly conserved among all the quasi-species that we analyzed from our local strains. So after infecting the cells with the antisense, with the virus, in presence and the absence of the antisense molecules, and following the cells for 48 and 72 hours, the first antisense compound against the AUG initiation codon at 2 concentrations, 1 micromolar and 2 micromolar concentrations, could inhibit both the synthesis of plus and the minus strand. Actually, the virus, hepatitis C virus, replicates through an RNA intermediate known as the minus strand. So the presence of the minus strand indicates that the virus is replicating. So both the concentrations used, 1 and 2 micromolar, could inhibit completely the synthesis of both plus and the minus strand after 48 hours and 72 hours. The other antisense that we developed against the stem loop structure was not as efficient as the one that was developed against the cis-acting element. However, after 72 hours, both concentrations could inhibit the first antisense molecule. So both antisense molecules at the two used concentrations were effective against the HCV replication. And this is the real-time PCR demonstration of the intracellular quantitation of the virus to show that our RNA is intact among all the treatments used, and also that to show that the antisense molecules was selectively reacting with the HCV sequences. We used housekeeping GAPDH, glyceraldehyde 3-phosphate dehydrogenase, to check the mRNA levels for this housekeeping gene, and it was actually fairly fluctuating over all the treatments using either concentration of the two antisense molecules. It was ranging from 100 to 180 nanograms per ml in all the treatments, which means that our RNA preparations were intact, and the activity of the antisense molecules was selective towards the HCV sequence. Of course, there are lots of experiments that we are involved in right now to check whether these will affect cell cycle regulating genes and probably other apoptotic genes. So we will do lots of control experiments to check the effect of these antisense molecules. At this point, we do not know what to do with this. Hopefully, we will start moving toward trying to develop delivery systems or trying to collaborate with someone to use the most optimum delivery system for these and move a little bit from the in vitro situation to chimpanzee, for example, which is the only non-human host for the HCV. Some pharmacokinetic studies that these SODNs, when injected into the small experimental animal, within 2 hours, 20% go to the liver, which is our target tissue, our preference target tissue, and 80% will go to the other tissues. 50% will be degraded in 2 days. As far as elimination of these compounds, in the first 24 hours, 30% will be eliminated, 70% will take 10 days to be eliminated. Tissue distribution of the compound will was fairly equal between the different tissues. And this is good because hepatitis C virus is not only hepatotropic virus, it infects also the bone marrow and probably other tissues. Kidney also incorporates HCV. Skin may have HCV. Some other tissues. Brain has little amount of the injected SODNs due to the blood-brain barriers or whatever. But probably during the delivery systems, we can use targeting molecules toward preference tissues like liver or any other tissues. But the native SODN can be distributed fairly between the different tissues. So in conclusion, the translation of HCV proteins is regulated by both cis and trans-acting mechanisms via the 5' UTR region. Also, the natural mutations that we have seen in the healthy carrier patients within the 5' UTR surely interfere with the life cycle of the virus via inhibition of efficient translation of HCV polyprotein precursor, and eventually they will affect the synthesis or the expression of the structural as well as the non-structural proteins. Therefore, the designing of antisense SODN compounds against the 3D stem loop and the AUG start codon may be efficient in HCV control in vitro and probably later on in vivo. Thank you very much.
Thank you very much, Dr. Mustafa. May I have the first question from a clinician?
Okay.
How do you think tissue engineering served in such a big problem in Egypt?
I just told you a few before I get to the stage here.
Okay.
We would like to collaborate with someone who will help us in developing delivery systems. Either we use the antisense or we use the small RNA interference or small any other DNA. The traditional drugs are not as efficient, and this is for sure, and this has been proven everywhere in the world. The response rate and the sustained response rate for sure does not exceed 50%. So we'll have to look for an unusual therapeutic option. So this is our effort to design or to try to look for the most important sequences within the HCV genome that we can design anything against it. There are several people who are working with the anti-proteases or anti-helicases or anti-polymerases, but we believe that from what we read, that the DNA therapeutics is, in our opinion, is the best way.
Thank you. We have the microphone, please. Microphone.
Can you get mine?
Okay. One moment.
I'm sorry to speak again, but I think this is brilliant work, badly needed in this country, for just because of the fact what you have just said, that this is a major, major problem, and therefore, please accept my humble congratulations on a brilliant work. My question is simple one. You already talked about the distribution of the virus in different parts of the body, and
The use of the SODN, of the
the
antisense.
ODN, but does that mirror?
But also the
Does
virus,
that mirror the HCV? Do we know where it hides? And in your delivery, what are you going to target? I mean,
Right.
The immediate thought is you want to go to the liver, but if it's hiding somewhere else, you
It
want to
hides.
target.
Yeah.
Where does it hide?
the largest extrahepatic reservoir is the peripheral cells, peripheral leukocytes, T and B cells. Probably the bone marrow. Many people have shown that bone marrow incorporates virus C, spleen. I didn't go over all the reports, but it seems that it is not only a hepatotropic virus. It can affect at least 5 different tissues in the body.
So that you're going to target that
Yeah,
in your delivery system.
yeah.
Thank you very much.
Thank you.
Thank you.
If you see this health carrier, follow them as any of them involve a critical stage of the virus for all their
Can you raise your voice?
Yeah, I got the question, yeah. Yeah, I got the question. The question is that what are the fates? What is the fate of these healthy carrier patients? I'll tell you a story about one of these patients who has been infected with HCV for 10 years. And he has been always like this, following this criteria. Normal enzymes, normal liver architecture, everything was normal. The core antigen was very low, or sometimes was undetectable. Extremely low viremia. He was well compensating the status. This was for 9 years. The last year, which was the 10th year of the infection, he managed to resolve the infection. No infection. And the patient was completely healthy. The only marker that he was that indicates that he was an HCV patient, that it was the HCV antibodies. That indicates that he has been exposed to infection. But he doesn't have viremia, he doesn't have, everything was normal now, and he managed without any interference with the drugs. And I think this is the patient that I have shown the mutation, the double mutation in the 3D structure of his iris region. So most of these patients, if we do not interfere with them, some of them, I don't have the statistics for this, some of them will resolve the infection.
I really do appreciate the effort done here for such a problem. Would you like to ask a question? Any comments?
Can I ask another question?
Please.
What other clinical categories are you targeting then? The severe ones? Can you still cure somebody who has liver problems and going down the drain?
Non-viral, you mean? Or
With any of these tragedies, or once you start having the pathology, is targeting the virus still a viable scientific strategy?
The only trial that we have done with these compounds is with the HCV, because there was emerging needs for this.
Yeah, I'm talking about HCV. Once they have liver dysfunction.
Right.
Where you have other strategies.
Okay. So what they usually do with these people, they give them the regulated interferon plus liver. I'm not a doctor, but the physicians usually provide the standard therapy, which is the regulated interferon plus liver.
But in the future, you think your strategy will have these patients as well?
We hope, yeah. This is why, we hope to. Actually, it is this work is at infancy. We have investigated different mechanisms for these healthy carriers to try to search for some light
to
get into. So we found that some of them they have different immune complexes, and we managed to also to develop a neutralizing antibodies that could interrupt the viral attachment to the cells in vitro, and we could block the viral entrance by 70% efficiency using neutralizing antibodies against highly conserved. The problem with the HCV is that, Dr. Magdi, is that it is a very mutating virus, very highly mutating virus. And the region that is supposed to be the most optimum region for developing vaccines or therapeutic antibodies is located within the envelope glycoproteins, which is hypervariable. So you cannot find any two quasi-species looking alike. So the idea is that to look for highly conserved areas, and it should be more or less exposed to trigger the immune response. So we could characterize a highly conserved decapeptide within the envelope E1, which is a little bit hidden compared with the E2, which is more exposed. And we use this to develop antibodies in experimental animals. We use these antibodies in vitro to challenge the viral entrance, and we monitored this by flow cytometry. We found that it really challenges the viral entrance into the cells, which means that if we humanize, or if we use the humanized counterpart of this antibody, we probably also have another option for interfering with the infection.
And you're pessimistic about vaccine?
I cannot tell, it is. I know that many people are working hard on this, especially in the United States and in Japan. Our little resources may, handicap us a little bit to continue. But the reason why I have been here in this workshop, probably to find someone to help me to continue to the next stage, trying to, to deliver, or to find a suitable delivery system for this.
No, we appreciate the effort you've done for such a thing. Please.
I got a question, Professor Dr. Ya'oub, about the other strategies. It's a real problem because not only hepatitis C, first cause of liver cirrhosis in Egypt is bilharzia, second is hepatitis, third is combined bilharzia and hepatitis, so it's a major problem, and fourth one is cardiac cirrhosis. One of the other alternative strategies for treatment is the use of stem cells. There is a team in Cairo University who take the autologous hematopoietic stem cells from the anterior superior spine and separates them and prepares them with hepatocyte growth factor, and then they inject it photographically guided to the spleen. They have finished the animal safety study, and they are going on with the clinical one. The clinical one they have studied, they had 10 controls, 10 cases with a smaller dose of 150,000 stem cells and 6 patients only with 2 million stem cells.
Delivering HGV.
No, delivery of stem cells for regeneration of hepatocytes for end-stage liver disease, and of course this does not help at all in solving the problem of portal hypertension, but it solves the problem of the liver cell failure. The comparative study is the control 8 out of 10 died in 6 months, while at the smaller dose only 2 died out of 10 after 6 months.
Thank you, Bruno. Thank you.
I want to ask, all your cases with the subclinical or with the carrier status had the same mutation in the virus, or some cases without the other effect? Did you study the other in the host immune system to see if there are other factors related to the host rather than to the virus in making the infection milder?
Okay. Thank you for the question. Regarding the first part of the question, we have analyzed the IRES region in several patients, and in almost 50% of these patients we found mutations, not only in the 3D, probably in the 3C or the 3B. And this, it is a pool of trans-acting factor interaction with the viral genome to modulate the rate of gene expression and translation of polyprotein precursor. So there are, in 50% of these 50 carrier patients, there are mutations within the iris region, not exactly the same mutation, but definitely if we do the software analysis for the sequence, it will affect the trans-acting factor binding to the stem loop structure. So this represents like 50%. We didn't do statistics. We did like 10 patients so far. So the other factors that are probably the immune system, this needs a whole lot of studies to analyze the difference in the immune system. Many people here in Shams University are working with the T cells and the B cells. It is not our specialty to work with the immune system or with the immune background of these people, but we know that the immune system may play a role in the control of the aggressive infection or aggressive disease in this particular group of patients.
I think we're running out of time, just to keep the schedule. Thank you very much for this. It's an area of good research to go on. We have been distributing the abstract for the special presentation that will be given, inshallah, Friday at 10 o'clock in Alexandria. So this is a